And today's speaker is Dr. David Held. He's a professor in the department chair of entomology and plant pathology at Auburn University. Dr. Held works on ornamental production and turf plants, nurseries. And today he's going to talk to us about managing borer insect pests in nurseries. All right, David, so I'm going to stop sharing and let you take it.
Sounds good. All right. So hopefully you're seeing the right view here of my title slide.
I think we're seeing your presenter view.
All right. How about now?
That's it.
All right, good. All right, so appreciate this opportunity to talk to everyone. We are going to talk about borers today. I do have a QR code here for a publication, but when we were pre-gaming here, I just dropped it in the chat. So you're going to find 3 resources in the chat. I encourage you to reference those if you're interested in this ACES publication on borers or publication on carpenter worms, and a review article that I think is pretty well written on ambrosia beetles. So let's just start out first by, I'm an insect sympathizer, right? So borers are not evil inherently, right? They do have an ecological niche. First off, it's really cool that they're developing, and animals developing inside a plant. We can all accept that as being really cool. But the thing that I think is their niche really is to remove weak trees from stands. All right, that's really their role. So just keep that in mind, because that's really important. That niche is important to think about when we apply this to how do we manage these issues, or how do we try to get inside the head of these borers and figure out a way in which to effectively keep them from ruining trees in nurseries or landscapes. So with all the borers, we basically have chewing mouth parts. That's in the larval form especially, but most of them in the adult form as well. Now, borers will be inside stems or branches, and usually that location where we find them is pretty indicative and can help us get some insight on what species it might be. And they form these galleries, and you kind of see the picture there of a gallery. The galleries are essentially mines underneath. So if you can imagine one of the life stages, most of the time it's the larval stage, is creating this pattern underneath the bark in the tree. As it does that, it's disrupting obviously the vascular tissue, which is on that outside part of the tree, which further weakens the tree and often leads to death. So we have 4 orders of insects that are wood borers. So it's not just some homogeneous group of borers, right? It's a lot of diverse insects. And for the purpose of our presentation today, we're really going to focus on just the wood boring beetles and the wood boring caterpillars in Coleoptera and Lepidoptera. You really don't care about Hymenoptera as wood wasps unless you're managing a pine plantation. And typically we don't have issues with the borers that are flies or wood boring maggots in our woody plants. So let's jump in and talk about some of these beetles, right? So the beetle borers, again, this is probably our most diverse group that we're dealing with. They're going to have these hardened wing covers and look very beetle-like. If you've ever had an entomology course, you remember those hardened wing covers are kind of key identifying character for beetles. Okay, we're going to talk about 3 main families though within the beetles, and how really to get some insight on what those immature stages look like in the tree to family. We can't really get them to species from the larval stage, and then talk about what some of the adults look like, because it is the important thing about the adult is that's what you're going to be catching in a trap, and trapping is really important. So these are our 3 families: Buprestidae, Cerambycidae, and a subfamily of snouted beetles, which include the bark and ambrosia beetles. So when we talk about these, what's really troubling for some folks when they think about this is trying to sort out these names. Wait a minute, you're calling it a metallic wood boring beetle, but it's a flat-headed borer. Yes, because we have a common name that describes the mature stage that's different from the common name that describes the adult. And that could be one point of confusion. So just keep that in mind, especially when we're talking about these beetle borers. So if we jump into these metallic wood boring beetles, they're actually quite beautiful. Well, I had a grad student that just finished up a project on Pests of Alabama. So we know quite a bit about this complex that attacks ornamental trees, at least in the state. Have a good feel for it. So these buprestids are really interesting in that their body is kind of cylindrical. So I want you to look at a couple things on this shot. Look at the bottom picture there. And if you can kind of think about these beetles as being kind of rounded on the top and flat on the bottom, because this exit hole is so very characteristic. If you ever see an exit hole that looks like this on a tree, the thing that emerged had to be in this family. Okay. This is the only family of wood borers that actually makes that type of exit hole that looks like a D, because the shape of the adult beetle when it chews out is flat on the bottom and rounded on the top. So we've had a couple of invasive beetles in this family get in. Emerald ash borer is a good example, right? So some of you have probably heard of emerald ash borer. We don't have a lot of ash trees necessarily in Alabama, so this was not the issue that it was for Michigan, Ohio, and elsewhere. But this brings up a good point for us to try and understand a little bit about the subfamily of these beetles and where the groups fit in, because you're like, okay, emerald ash borer is related to this ugly brown one over here that's a flat-headed apple tree borer. And yes, but they're represented by two different subfamilies. So the emerald ash borer subfamily, Agrillinae, all of them in that subfamily tend to be more parallel-sided, and really essentially their body is not really wider at their wings than the outline. All right. So again, that's what I mean by parallel-sided. Essentially, the body is no wider than the head. All right. And so if you look over at Buprestinae, now this is where our flat-headed apple tree borer subfamily fits in, and a couple of the really important ones that attack pine trees in Alabama. And what you can see is kind of, you've got the head, and then the body then right there where the wings meet on the thorax flare out. So you actually get the widest part of it right there near the head, on that part of the thorax where the wings connect. It's not parallel-sided. Okay, so that's just a good way to kind of separate those two to subfamily. We do have, you can't really use coloration. I wish I could tell you like, oh, well, all those are real shiny and the other ones are not, but because we do have pretty ones in this entire family across both subfamilies. But these are some pretty cool beetles, and just wanted to show you something about what a character you might see if you collect them in a trap. Now, if you were to dig around in an infested tree and find the larva, you would see that these kind of get this name of being flat-headed borers. And it's not really the head that's flat. The head's actually inside that part of the thorax. It's retracted in. You can't really see it. The mouth parts are just barely sticking out so it can feed. But that is a part of the thorax that actually is expanded and enlarged, which gives them their common name, flat-headed borers. And you can see that on the emerald ash borer larvae in the bottom and in the sort of line drawings in the top. Now, the other thing that's in this family that's kind of unique, we don't really see these things called pseudocerci or Urigomphie in some of the other borers. And what these are basically, whenever you hear cerci, think pinchers. Okay, you've probably seen an earwig and seen those pinchers on the end. Those are basically sclerotized cerci. Cockroaches have segmented, more softer cerci, but these are real common in insects. And they're not, they're not there to necessarily pinch, but essentially this family, if you find them in a tree with that flat head, you'll see on most of them that they will also have these pseudocerci. So the common species really tend to be in Alabama, tend to be in this what we call the Chrysobothris femerata complex, and in lay terms, that's basically the flat-headed apple tree borer complex. And this is a group that would attack maples and some other hosts. They have some basically hardwoods. They have a hardwood host range, And that's pretty common for most of our buprestids anyway. We do have a few that just attack pines or needled evergreens, but for the most part, these are going to attack hardwoods and have a wider host range. So if we go back to that separation I mentioned earlier about the one subfamily that these Chrysobothris are in versus the subfamily like emerald ash borer, we find that the flat-headed apple tree borer complex is actually earlier active in the spring. We tend to get more of the groups with the emerald ash borer being a little more summer active. As I mentioned before, I had a student finished up a project with the flat-headed apple tree borer complex. We trapped 10 species of Chrysobothris statewide. Interestingly enough, we were like, well, let's rear them out of trees. And Jeremy helped us with this down in Mobile. And we tried to put out trees and get attacks on these trees to find out what species that we catch in traps are related to attacks on trees. But over 3 years, we never found a single tree, even ones we've cut down around Auburn with my chainsaw. We never reared an adult successfully from any of those trees. And as I mentioned before, we can't ID the immature stages. We have to rear out the adults and then identify them. So that's a bummer. If you've got infested trees with some of these flat-headed borers, I really would love to have them and rear them out just to kind of get a better feel for what is attacking trees in Alabama, because we failed miserably at that over those 3 years. That flight is starting right now, especially for that flat-headed apple tree borer complex. If you're a nursery producer and you have maples in particular, and I don't have time to go into those susceptible maples, but that's online. We can find that for you if you need some help with that. But maple producers, especially in the spring, can have problems with attacks on these trees, especially as they're putting out new bare root material in the field. So that flight's going on right now. It'd be something to be vigilant out there, either trapping or monitoring for. Let's go to the other subfamily. These are the longhorn beetles that produce round-headed borers. Huge beetles. If you ever see these come to a porch light, you're like, I didn't know insects got that big, but they do. So longhorn beetles, those antennae are just really long. That's where they get their common name, usually twice as long as the body. They get this round-headed larval appearance. It's not flattened like the flat-headed borers, so you won't see that same thing. They don't have those pseudocerci. A lot of people have difficulty saying, is this a round-headed or a flat-headed borer? But again, you can use those pseudocerci or Agamphi as another way to kind of separate these families if you find the immatures inside a tree. Again, the tree is often very useful also for identification for these round-headed borers. Just to kind of give you some idea with that, you know, if you have, if you were to rear out a cerambycid, a round-headed borer, collect one from a dogwood, it's more than likely going to be dogwood twig borer. And so you can kind of see those host associations, red oak borer and others that are associated with certain species in this family of beetles. I want to wrap up with a third species, third family of beetles, actually a subfamily of beetles. And most of these here, we don't really think about in nursery production because they're pine. And so our forest entomologists are usually concerned with those, with the exception of this group that are called the ambrosia beetles. Now, the ambrosia beetles, the one thing I want to convey to you is that these are really tiny beetles. As my eyes age, I can't really see most of the characteristics on these anymore. They have to go under a microscope or I'm not going to be able to do any kind of ID with them. These are the smallest of our wood-boring beetles. Let me give you some perspective on that. So these are the 4 common species that would be in southern nurseries and landscapes. Nestis mutilatus is the big one. And not only is it large in size, but the back end looks like someone took a hot knife and went through butter and just cut the, basically cut the abdomen off, essentially, because of the way that appears. So the front end of the beetle looks bigger than the back end here. And that's really unusual for these ambrosia beetles. But we have then 3 other species in the genus Xylosandrus, Crassiesculus, Germanus, and Compactus. Of those, probably Germanus and Crassiesculus are most commonly reared from damaged trees across the Southeast. Now, again, the small size makes these really challenging to identify, but then the head is hidden under the body. They've got these tiny little features that we then have to look at, like this part of the wings called the declivity. We've got to look for hairs on them. So hopefully this gives you a little bit of appreciation for how difficult it is to separate some of these things, especially when you see them in a trap. Again, we'd have to get them off the sticky trap or out of the lingered funnel trap, bring them into the lab and put them under the microscope just to be able to separate these 4 species that we have commonly in nurseries. It's not just something you can walk up on a trap and say, you have Xylosandrus crassiesculus. It's just not that easy. The larvae here are grub-like, so they'll be kind of C-shaped, just a little weird, and of course, very small. They're also legless, like we see with many of our other borers. And what's interesting about the ambrosia beetles that, that we don't see with the bark beetles is the ambrosia beetles are not really eating the tree. They're carrying around ambrosiella fungi, and that's what the larvae are feeding on. So they're fungal gardeners, and they're feeding their immatures on fungi, which makes perfect sense to me because fungi are more nutritious than tree tissue, but that's why they carry them around. All right, we're going to move out of the beetles, and I want to talk about our wood-boring caterpillars, and just really 2 families here. But one thing about wood-boring caterpillars, if you were to dig in the tree and pull out some larvae, you're like, how am I supposed to know if this is a wood-boring caterpillar or a beetle? Well, one thing that the wood-boring caterpillars have are these prolegs that have crochets. Now, I don't know if your aunt, your mom, your grandmother ever crocheted, but I mean, growing up, I always got an Afghan from my grandmother. It seemed every 4 or 5 years for Christmas. And that little crochet hook is the perfect description of what these things look like on the prolegs of these caterpillars. You'll never see those in a beetle borer, but they're just these tiny little hooks, and they're not tiny. I mean, they're obviously you're going to need a hand lens to see them, but you won't need a microscope to see them. So that is a good character to kind of separate this order of borers from all the beetles. If we look, we've got 2 families here, and I'm just going to use common names here. The clear wing moths, they get their name because they really don't have scales on the wings, which kind of one of our defining characters of moths and butterflies. But most of them also look like wasp mimics or bee mimics. So they basically have these very long, kind of narrow wings. You contrast that to carpenter worms, which have a typical moth look to them with scales all over the wings, and they tend to have very wide wings. So, and be very characteristic when you find them. So the clearwing moths will, unfortunately, I can't tell you the flight period because it'll vary by species, whether you're talking about dogwood borer, lesser peach tree borer, or banded ash clearwing. They're going to have different flight periods. So I can't make a big generalization, but Jeremy and I and some of our students have studied the carpenter worm moth flight in Alabama for a number of years. And we know that flight runs basically from March through July because we put pheromone traps in nursery with nursery collaborators and figured that out. So that's the period when we'd have carpenter worm moth activity. So if we talk about the biology, there's a real big difference with the biology of borers. So obviously there's differences with their taxonomy, but for the most part, all these borers are going to lay their eggs on the outside of the tree, the surface of the tree, and then that immature stage is going to chew in. Now, the exception to that would be ambrosia beetles in nurseries. So the ambrosia beetle female, she goes in and excavates the gallery and then lays eggs in the gallery because she's going to introduce that ambrosia fungi for the immatures to eat. Okay, so she just can't leave them on the surface and be like, okay, find it and eat. She has to put in the fungal garden too, so that those immatures can eat the fungi. So that hopefully will make sense ecologically there. And this is why we see frass sticks associated with ambrosia beetles in nurseries, because when that female invades, as she's digging the gallery, she's pumping that tree tissue in her poop right back out. We call them frass sticks because it's a mix of sawdust and the female's poop. And then as she excavates that gallery, she'll lay eggs, introduce the ambrosia fungi, and then you can kind of see the larvae that are developing in that cross section of one of my students cut open there in the bottom picture on the left. Okay. These are incredibly female biased populations, 10 to 1. We almost never see males outside the tree, but they produce a lot of females. So we are in the flight for Xylosandrus crassiesculus right now, and other Xylosandrus in nurseries are going to be coming on really soon. I tend to use peak bloom of our calorie pairs in the landscape as kind of a trigger to tell me that it's time to put out traps. that we're going to get flight. So really, when you start to see those coming on is a time when you might want to get your traps out so you can maybe catch that first peak. But that is a really good plant phenological indicator for activity of these ambrosia beetles in nurseries. And again, they overwinter in all stages in the tree, especially the adult stage, so they can just pop out. The first generation in the spring is very synchronized. We get a lot of adults in a real short amount of time. And then the rest of the year, it's these very asynchronous generations. So they'll be putting out a few. So we really focus on trying to manage the spring generation more importantly than some of the others. All right, so let's move out of biology, taxonomy, and let's talk about the nuts and bolts of tree diagnostics. So with borers, we tend to see that they weaken the trees. And again, or the tree could be a little weakened from some sort of injury, lightning strike, drought, you name it, bad pruning if you're in my yard, all right? And that could provide an opportunity, cracked bark or something, for that female to lay her eggs near that to allow that larval stage to get inside. We know especially that water stress, whether that's drought or even flooded conditions, with some of the ambrosia beetles are really important indicators of or predisposing even some of those trees to attack. The bottom, both those pictures are from places where I've reared beetles out of trees. The top one happened to be a buprestid that I reared out, and the bottom one happened to be an ambrosia beetle where we saw delayed leaf flush in a nursery. And come to find out, those trees are being attacked by Nestis mutilatus. In fact, there in fact is the couple of those D-shaped exit holes for those flat-headed borers that we see associated with that tree that was damaged. Now, it was producing these really weird frothy bubbles, and I had to ask a plant pathologist about alcoholic flux at that time. But that was another one of those sort of tree diagnostics that I now associate with either borer attack or being predisposed to borer attack. We also have signs on the tree themselves. And so let's unpack these. If you see pupal skins, if you look at the top image, a pupal skin sticking out, it absolutely can only be a caterpillar. Why? Well, because the adult moths, and you know this about moths and butterflies, they don't have chewing mouth parts. They've got that kind of fire hose they unroll and suck up nectar, right? It's elongated proboscis. And so the immature stage, that larva has to chew that hole, that emergence hole, and then that pupa just wiggles out, okay, and leaves behind the pupal skin after that adult moth emerges. 100% diagnostic. You always know you have a caterpillar borer if you see that. Now, emergence holes with the beetle borers are going to be one of our best indications. I've talked several times about this D-shaped exit hole as being indicative of flat-headed borers. And then we generally use hole size kind of as to separate the round-headed borers. And we know those are the largest, and the small ones being the ambrosia beetles. They tend to be our very small and very numerous because I did mention before they attack in mass on these trees. And you can kind of see there, that's one of those Nestis mutilatus that was being pulled out of one of those trees from that nursery I showed you previously. All right, let's wrap up this with some management, okay? So I want to talk about these three areas in particular. You've heard me talk about timing and biology. So trapping is going to be our proxy for timing, okay? Without trapping, we don't know timing. Then we're going to talk about tree health, and I've thrown some of that stuff in there a little bit. And then I want to park out on and talk about this insecticides and application, and sort of pause there and let some of that stuff sink in and think about how you can manage these borers. All right, so as I said before, trapping is our proxy for adult emergence. We really don't know when the adults are emerging unless we have a trap out, because they don't check their watch and they don't look at a calendar. So it's going to vary based on temperature every year. And we do see that variation quite frequently. So we commonly use traps. You can use plant phenological indicators too, like I mentioned before, with the peak bloom of the calorie pears, right? So as you trap, you might be able to associate that with, okay, I always pick up my carpenter worm moths when this is blooming, or I always see Nestis mutilatus when this is blooming. So you may not have to trap forever. You may make those associations, and again, may only have to trap for 3 to 5 years until you build those associations. But pheromone traps are very common and very specific. Then we have these ethanol lure traps and visual traps that tend to be less specific. Um, and I want to show you what this ethanol baited trap looks like. This is a lingard funnel trap that was in a nursery in May of 2018 in a nursery we're working in. And when you look inside there, that's a basically probably like a 3-day catch of Nestis mutilatus. And that's what that larger ambrosia beetle looks like in the bottom of a lingard funnel trap. But again, that generic trap will catch any type of ambrosia beetles. Only pheromone traps are species specific. Okay, and so we tend to only have those for our caterpillar wood borers. Okay, for Lepidoptera. These other traps, the purple panels, the purple poles, they're going to get a lot of what's called bycatch. And so you can get false positives because there are a lot of beetles that would get trapped on this inadvertently, just flying, and a lot of other things that could get stuck. So you're going to have to refer back to some of those characteristics I gave you of the adults before to make sure that you're not looking at a click beetle and saying, I have buprestids active in this nursery, I need to go spray, because that's wasted money. All right. The last thing about trapping we know is that a lot of times the species that get stuck on these traps are not always the species that we rear from trees. Okay. And again, I'll just go back to that buprestid study my student just wrapped up. We had 10 species statewide. We never reared a one of them from a tree that looked like it was infested. Okay. And if you look at that same trend with ambrosia beetles, we see that as well. So tree health, and what do we do with trees once they're infested? Well, first off, we got to prevent some of these stresses, trunk injuries, water stress whenever we can, okay? Because those are, again, precursors to some of these attacks. The sort of running wisdom, not a lot of data with it, but the running wisdom is you leave the tree in place and you only cull it after the adult flight. So essentially you're creating a trap tree. We want to have as many borers come to that one tree because we're going to take it out anyway. So let it just get loaded up with borers. All right. And you might think that's kind of counterintuitive, but that's kind of the running wisdom right now, especially relative to ambrosia beetle management. And then what happens with that tree when you pull it out of the nursery? Well, we go and we pile it up somewhere. Well, you really should chip it or burn it, because just look at the data from this carpenter worm trap that we have. So rather than count the moths, I think visually it really shows. We put pheromone traps out in the nursery versus at the exact same time we had them next to the cull pile from last year in a nursery that was really having issues with carpenter worms. And that cull pile is essentially a reservoir for the next generation. So you really need to deal with that, especially with these borers, because they can continue to develop complete development and emerge from that cull pile. All right, so relative to insecticides, our last big topic, I want to talk about timing relative to trapping. What are we going to expect from our chemistries? And then some work we did with application methods and residues. All right, so timing, we typically, and the common wisdom has been, you apply some sort of trunk spray 2 weeks after the first capture, or within 2 weeks of the first capture, and you reapply, because residues are going to wane. And in spring, we're always going to get some rain. So we want to make sure that we keep a fresh residue on that trunk to prevent the tree from being attacked. That's the end goal. All the discussion about biological control, I'd love that in context of other insects, but biological control would be attacking the immature stage, which is already in the tree, which we don't really want in a nursery situation, because that's just going to weaken the tree. Okay. So we really want to intercept these borers with insecticides and try to get that residue on there to either kill that adult female before she goes in if she's an ambrosia beetle, or kill those larvae. Permethrin and bifenthrin are by far probably the most common. We still have labels for some of the older chemistries, the organophosphates and carbamates. Now, there's been some good work with ambrosia beetles with the discus approach and using that product in nurseries for ambrosia beetle management. And it shows multi-year, which is really uncommon to get multi-year control with some of those products. But we also see some of that, a little bit of that benefit with flat-headed borers and longhorn borers as well. Now, if we look at the moth borers, we actually have access to some other chemistries other than things like permethrin or bifenthrin, because we're dealing with a caterpillar, and we have a bunch of caterpillar-specific products like spinosad or acelyprin. These materials are really were developed to target caterpillars. And once you spray them out there, they can be very effective. So again, we have a lot of data with different products looking at ambrosia beetles. They tend to be our lab rat for borers because we can manipulate attacks and we can sort of predict when they're going to show up. So this tends to be our lab rat for borer, a lot of borer projects. We know that kaolin clay, which you see on the white covering on the trunk here, or alone or with pyrethroids, it can be very effective. We did this for carpenter worm, showed reduced attacks. It's been done with ambrosia beetles. I've not tested limonene, but I've seen it in the literature suggested that limonene is a repellent for ambrosia beetles. And then there is some work with insecticide treated nets, also with ambrosia beetles. And this is not going to ever work in a nursery, but if there was a valued tree in a landscape that needed to be protected, perhaps like our, I don't know, our tumors oak trees when they got put in, if we didn't want to spray them all the time, we actually could have wrapped them with these insecticide treated nets. And that would have prevented some attack, at least during the initial transplant when they were under perhaps a little bit of stress. So application methods, basically almost universally, I think when I see in nurseries are air blast sprayers. Now, in research though, when I go out put out a trial, I'm using trunk sprays. I'm getting right up and spraying. Now, it uses a ton of labor. I get that. And, but it also uses less insecticide. And we use less insecticide because we're not blowing it over all these unintended surfaces in the nursery as well. So again, I get that it's very labor intensive, but you could use less insecticide with those types of methods. Again, that's how we do our research studies when we want to look at the application of these materials to the trunk. And residues are so key. And this is why earlier on I talked about reapplication. Almost all the borer recommendations have some sort of 10, 14, up to 17-day window, depending on it. And that would be shorter if you have a rain event or if you have overhead irrigation. So again, but residues on the bark are really important during that period when adults are active and laying eggs, or in the case of ambrosia beetles, attacking the trees. I want to wrap up by talking about one of our case studies that we did a number of years ago. During COVID, we got permission to keep this going because it happened in the spring, but we collaborated with a nursery to look at onyx, which again, synthetic pyrethroid, applied in the nursery. And then we took a chainsaw and cut the trees up. And we had the lower, middle, and upper trunk sections to basically ask, what about residue deposits on these just under 2-foot sections of tree that it just were exposed to an air blast sprayer? Are we delivering the residue we think we are? All right. And the answer to that is no, not even close. So the maximum residue we had on the trunk was 11.6 parts per million. If you calculate what the tank mix was, it was about 375 parts per million. So we're not really delivering a whole lot out there. And as you can see, if you look at, there's tremendous tree to tree variation, and we're tend to hit mostly, if you look at kind of the upper parts, kind of the middle and the top, they almost always have higher residues than the bottom. And unfortunately, the majority of attacks from these borers are coming in the bottom 2 feet of the tree, which would have the lowest residue if you're using something like this air blast sprayer. So again, we have some issues where we could have breakthroughs in treatment, not related to insecticide resistance, but just to application method. We need to make sure we get deposits in this lower part of the tree, because if we're bed hedging, that's where the attacks are going to come. Okay. What's still unknown is exactly how much residue to benchmark those residues against what it takes to kill these insects. Don't know that. That's still one of these things I wish we had those data. That would be really important to know. All right, want to acknowledge our funding sources and all those that helped out. We had a lot of support from the USDA, National Institute of Food and Agriculture, to do a lot of this work. I have put the resources into the chat for you, but here's some of the ones that are dropped there. So I'll pause right here, maybe see if there's any questions, and point to my email there if you need to reach out and get a hold of me.
Dr. Hill,
Yeah.
I was looking through all of those, and the only thing I did not catch hosts for was the ambrosia beetle there.
Yeah, so ambrosia beetle has preferences, but it doesn't really have um, host specificity because again, it's not feeding on the tree. So remember, I went pretty quickly by that, but that female carries the Ambrosiella fungi. After she makes that gallery, she starts a fungal garden. So as long as that fungal garden can grow inside any tree, that can be that could be a host for ambrosia beetles. So the common ones I see attacked, golden rain tree, really common. Oaks are being attacked pretty commonly. Uh, Crepe myrtle under certain situations. The summer generations like to hammer crepe myrtles, especially under flooded conditions. But that is a challenge because there really isn't a good host association because they're not consuming the wood of the tree. Does that make sense?
Yes, sir. Thank you very much. I was just looking for a place to start.
Yeah, that's a good review article too that's in the chat. That
I
one
appreciate it.
some of that as well. Other questions?
Hey, David, it's Jeremy. What so you said you got a pretty wide range for the bifenthrin or the Onyx Pro with the when you were looking at residues. And did how does that kind of relate to like the an LD50 or something?
I
Do you
wish I
know like
Yeah, I wish I knew. We don't have
yeah.
those um you know that information for the range of like Nestus mutilatus and Crassiesculus. We don't know if 3.6 parts per million can kill them in 12 hours or if they just laugh at it and walk over it and still attack the tree. Uh, um, yeah, we've tried to get at that a little bit with changing rates with bolt tests in the field, but we don't get consistent attacks enough for anything to be definitive. We tried that 2 separate years. Uh, so what it means is we're going to have to collect a crap ton of these in a lingered funnel trap, bring them into the lab, and just do straight toxicology with it. Is what I think we're going to have to do if we want to get that answer.
Yeah. I think one of the take home messages from that was where how you position uh, the output on your air blast sprayer. So you're saying we need to be pointing them lower uh, and not really worrying about the canopy so much as especially that bottom 3 feet or so.
Yes. Yeah. So that bottom 3 feet is probably the most critical zone for attacks for the common borer species that we have. Now I've seen Nestus mutilatus from branches in nurseries, but it's, if you were to map the distribution in a tree, it's going to be mostly the lower part of that trunk being attacked.
And sorry, I'm hogging up all the questions here, but could you talk a little bit about water stress and ethanol?
Yeah. So two things that we know is that water stress can either be too much, too little relative to borers. Okay. We also know that it really water stress doesn't mean a whole lot for the lepidopteran, the wood-boring caterpillars. So this is primarily a really important cue for the beetle borers. So if you have basically if you have drought, the tree is not able to conduct a lot of fluid, and in some cases they could even flush out certain borers like the ambrosia beetles. If there's enough fluid, they can flush them out. We see that in some trees, but that's related to defense. We also know that under overwatered situations, there's really some really good work that came out of Chris Ranger's lab and the group at Ohio. They basically dropped redbud trees or dogwoods into buckets with water. So basically they were flooded, and they were able to manipulate the flooding level to show that there's actually a flood threshold by which ambrosia beetles attack those trees, particularly those trees that are susceptible in particular to flooding.
Gotcha.
And they're just producing ethanol. And that's why the ethanol works so well in the trap, to tie it back to the last part of your question.
Dr. Hill, this is Steve Larson. I'm the technical rep for InView, and we're the company that manufactures Onyx and Talstar. It's a recent acquisition for us from FMC. And so I'm kind of learning about the use of onyx in the marketplace, and your talk was great. Really appreciate it. Do you know if there's been any meaningful work done with adjuvants in combination with something like onyx, either as a sticker, kind of a general use term, or as a penetrant that might help out with not only the efficacy of the product, but maybe might help the longevity of the product to remain on the trunk?
Yes, Steve, there have been, but it's all been bucket chemistry like experiments. And, just to unpack what I mean there, it's kind of crude, but so we mix it up, we spray it, and we might see fewer attacks, but we don't know why. We don't know if it's because of a penetrant. We don't know if it's because it's making that residue last longer, but we do see benefits of some of these various adjuvants when they're mixed in for application targeting some of these wood boring beetles in particular. But again, the question is why? Why does that work? And I can't find that in literature, but I can find plenty of insecticide experiments where adjuvants have shown to be more effective. Either spreading out that residue from a 10-day to a 17-day, or just a lack of attacks on those trees.
Sounds like an absolute I don't know.
There you go.
Thank you.
Mhm.
Hi, Dr. Help. This is Edgar Benson.
Hey.
Hey. Hi, you mentioned the use of the calorie pair as an indicator to begin placing traps, monitoring traps. But I was wondering, could a grower use that as an indication to start spraying?
So that makes my spidey senses tingle a little bit, Edgar, when someone wants to just use a plant phenological indicator to trigger sprays. Because I prefer to also verify that they're present in the nursery or the situation and not just assume they are. And that's what trapping gives us. It gives us confirmation that not only could they be in flight, but they are in flight where I do have susceptible hosts, if that makes sense.
Oh, yeah, understood. Yeah, thanks.
Mm-hmm.
So David, you can build your own ethanol trap out of a Coke bottle and
Absolutely.
stuff I've seen. But the best part about that, especially for the ambrosia beet, is it tells us when to start and potentially when we could take a break. I mean, if you
That's right.
stop collecting them, then it's like, okay, we can let off of spraying but you keep trapping until you come up with another adult, right?
Yeah. So what that means for ambrosia beetles is we have this spring window that's very active. So it could start anywhere February, March, and that's when we would get our first captures, and we would continue to trap, and then it's going to drop off. You won't see anything in the traps. We've done this. I had a grad student who did this in Ohio, and we did it in Alabama. It drops off like a lead balloon, and then that's where you start getting these really asynchronous generations. There's a few of them coming out, so we don't have the mass attacks, at least in nurseries. Now, if this was a landscape, if we were talking to landscapers, it'd be a little bit different story, because if we get heavy rain events, 4-6 inches of rain on a crepe myrtle and it's sitting submerged in that summer generation, it's going to be full of frass sticks. I mean, with like 75-80% certainty. Just because I've seen that happen. I rarely do things attack crepe myrtle, but ambrosia beetles love them in the summer, and they will mass attack that particular plant species. Why? Have no idea. To go back to Steve's thing, it's an absolute. I'm uncertain, right there, but I do think in nurseries at least, we want to make sure we're treating as long as we're still getting trap captures through that spring generation.
Thank you. Does anybody have any other questions?
Yeah, this is Brian Keller. Dr. Held, I really appreciate the information. I'm interested too, the discus, you mentioned multi-year efficacy on that. So and I guess I look at it a little different. I'm more of a, yeah, chemicals are expensive, but if a spraying saves one tree, then I pay for that spraying, so I'm willing to put more, put more chemical out. So I'm really interested in the multi-year efficacy because I mean, it seems like I never know when in the spring until it's, it seems like once I start seeing them, it's just they're just in mass. So anyway, I just would want to know a little bit more about that discus because that's and then could you maybe do a fall application or like like a January application? Do you think that would help, kind of get ahead of it?
So that work was done in Tennessee, and I could get out of my slides here in a minute and drop it in the chat if you want to read it. But that's the Dr. Oliver, Adesso, the group at Tennessee State did that work. And I think that they were really proposing a spring application, unfortunately, but they have considered the idea of, like, we don't have as much activity in the fall. Could we put it out in the fall? I'm not sure if Dr. Oliver has done that follow-up. He may have, just not familiar with it. But so that group actually did that research that didn't come out of my program. But I can drop that paper in the chat if you want to read it. So I don't have as much firsthand knowledge on it because it's not a study that we did. So I don't know the real answer to your question about fall applications, but it's really labor intensive though. I mean, they are applying discus, in solution to the base of all those trees. So again, you have some labor associated with that as well, Brian.
Yes,
How high, like whenever it looked like you weren't on the base of the tree, do you go up 2 feet or is it just like a 6-inch when you're applying that by hand?
Yeah, so in our studies, when we've looked at protectants for ambrosia beetles primarily, but also flat-headed borers, we're going up either to the first branch or that in that 2 to 3 foot high. So depending on what's there, we want to protect at least to the first branches. But if it's some sort of whip that doesn't have any branches, it's being transplanted in a nursery, we're going up to between 2 and 3 feet, and then to the ground and on all the way around. We're getting the whole circumference basically of that, all the surface area that would be exposed. That's how we do it for research trials.
Yeah, I understand. Thank you. I use a cannon sprayer, so like time wise, it's not a big, I can do the whole nursery in a couple of hours, and we just move everybody to a separate area, when we spray. So time using that method, but I do understand it's not as good, as doing the trunk spray. And just one more thing, I'm not trying to hog either, but we did use a penetrating surfactant this year, and that did seem to help. I mean, and I don't have data, but just from looking at it, it does seem to help.
Yeah, and there are data that support that in the literature, but again, we can't answer the questions I think that we're being asked about longevity or penetration, like where it's going and if it's affecting length of residue or why it's working. But we do have plenty of data that support your observations that it does work.
Well, good. Well, I think that's about all the questions we have. David, thank you so much for your time today. We really appreciate it. This was really informative.
Great.